I am Juliet Mazola, PhD student at the Center of Bioinformatics, Simulation and Modeling from Talca University, and I am part of the team working with Dr. Wendy González in the structural and functional understanding of ion channels. In this work, we are presenting a preliminary study that compares flecainide binding sites in atrial-selective potassium Kv1.5 and sodium Nav1.5 ion channels. These channels are involved in cardiac action potential, and they are important targets for atrial fibrillation, especially because Kv1.5 is mainly expressed in atria, and Nav1.5 shows different electrophysiological properties in atria than ventriculars. The use of atrial-selective targets decreases the incidence of ventricular proarrhythmia, which is one of the most relevant side effects in antiarrhythmics currently used to treat atrial fibrillation. Importantly, combined inhibition of both targets is predicted to provoke a synergetic effect, according to mathematical models of the human heart electrophysiology. To explore the potential similarities at drug binding sites of these two channels that could explain multi-target inhibition by a unique drug, we selected flecainide antiarrhythmic as reference. Flecainide is in clinical practice for recent onset atrial fibrillation. It mainly blocks Nav1.5 sodium channel, but also potassium channels, including Kv1.5. We took advantage that cryo-EM structure of flecainide in complex with Nav1.5 were recently solved, and putative important residues for flecainide interaction with Kv1.5 are also described from mutagenesis studies. No structure of K1.5 is available. Then we are using a homology model for this channel previously reported. Following this introduction, we will go to results. First, we perform ensemble docking using frames from 100 nanosecond molecular dynamics of Kv1.5 in its aper state, followed by clustering of ligand docked poses. We selected complex with the lowest energy score from the most populated cluster. This complex was compared to flecainide bound to Nav1.5 cryo-EM structure. Docking results shows that flecainide has a similar binding mode in both channels. In Kv1.5, the piperidine ring points to the central cavity, while the trifluoromethyl moieties are inserted into the subunit interface. In Nav1.5, piperidine ring also points to the central cavity, and trifluoromethyl moieties are inserted into fenestration. These binding sites were later compared using sitemap. Both binding sites show similar drugability score, and Nav1.5 shows a higher hydrophobicity that can be seen here in orange colors. These binding sites were further aligned by superposition of flecainide poses. We decomposed binding sites into 4 groups. 1, 2, 3, 4. By visual inspection and clustering of residues using K-means method. Importantly, groups in K1.5 show similar physicochemical nature with their counterparts in Nav1.5. That is, group 1 is hydrophobic in both channels. Groups 2 and 4 are mostly hydrophobic in both channels, and group 3 is mostly hydrophilic in both channels. Importantly, two residues included in group 3 that belongs to the selectivity filter in both channels forms hydrogen bonds with the protonated ammonium of the piperidine ring of flecainide in both channels. Geometric centers of these groups were plotted and can be seen here in the form of spheres with different colors. Distance between these geometric centers were computed along 100 nanoseconds MD simulations of flecainide complexes. Here we can see distance between this geometric center, for example, distance between geometric center from group 1,2, from group 1,4, and so on. In Kv1.5, we see that these distance are conserved along the MG trajectories, and a similar behavior is observed for Nav1.5. Also, mean values of these distances are comparable between corresponding groups in K1.5 and Nav1.5 channels. The results presented here are preliminary, but they give insights into the existence of a putative consensus binding site in K1.5 and Nav1.5 for flecainide. Hereafter, we are planning to extend our pocket comparison studies on MG trajectories of flecainide complex. I would like to thank to Fondesit for support our work, and also thanks the opportunity to present our results in this second Congress of Women in Bioinformatics and Data Science. Thank you very much.